Alright, guess what? It was a waste of time, and my lesson ends another 30 minutes later, so it's all good. Um, cha 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 cha. Okay, it makes me quicker, so I guess it's not so bad after all. Let's quickly just go over it. So, phase 3 now, 5,000 people being used. Let's say 5,000 people are being used. Everything is going more or less well, and the regulatory authority is happy. Okay. If they're happy, if we're able to find good results, if we're able to see that it's effective, if it's helping people without that many bad, unacceptable side effects, then phase three has now been phase three has now been successful. Now it has even a great. Now it truly has the chance. Now it has like a great, truly possible chance of becoming a useful medicine. Double trial, blind trials. I don't know what this is, but let's talk about it. Double-blind trials. Phase 2 and Phase 3 trials are normally carried out as double-blind trials. So within Phase 2 and Phase 3, we have what do we have? We have a greater sample size, and within these trials, we are doing it in a different special way called a double-blind trial compared to the Phase 1 trial where we are only using a very small sample size. Correct? This means that neither doctor nor the patient that the doctor nor the patient knows whether the patient is receiving a new medicine, a placebo, or a control medicine, right? So even the doctors don't know whether who is given what. Okay, this patients often appear to respond to a treatment because they believe that it will do them good. There's much that is not understood about how the mind affects the body. This response is called the placebo effect. This is a very interesting sentence that they used here. There is much that is not understood about how the mind affects the body. The mind does affect the body. The body may even become better because the mind feels better. Emotions truly change and shape how your body affects how your body gets affected by what you think, by what your mind says to your body. Because God knows how many hormones that produces just depending on what kind of emotions you feel that can change entirely how your body can function. Truly, it's crazy. So, patients often so this is kind of a more physical output. Patients often appear to respond to treatment. What does responding to a treatment mean? That it means that they respond positively. They actually react to the treatment. If I give them a medicine, the medicine will do something. It won't just sit there and do nothing. Usually, a medicine won't do something bad. Usually, the medicine will do either nothing or it will do something positive. Okay, and usually the treatment it does do something because it does do something, assuming it's positive, because they believe it'll do them good. So their mind thinks that it'll do it good. So then even the mind has a say or has a way of reacting to this medicine. This is called the placebo effect. Now we mentioned the placebo effect before, but now we're actually talking about it, how the mind affects the body. The use of a double-blind trial allows this to be measured. So the double-blind trial allows even the placebo effect to be measured. This is the point of the double-blind trial. Instead of a placebo, a control medicine, the best performing available treatment is sometimes used to avoid any patient being denied treatment when they take part in the trial. What that means is, instead of being a placebo which has no active ingredients, usually what's given instead of a placebo which is the control medicine, the best performing available treatment is given, so that no person is denied the treatment when they take part in the trial, even if it does something. Everyone has a right. If I volunteer for it and I get placed in a placebo group, then that's unfair because I didn't get the medicine. So that's true. So yeah, that's the alternative. And also we did talk about this in phase 2. So in phase 3, that I was right. This also happens, right? There are three things that can be given. There could be given the actual medicine that's being tested, the best performing available treatment, or even uh, the placebo. Okay, so Yep, that makes total sense. Phase three trials are used to confirm the effectiveness and safety of the new drug. As they do, of course, the number of patients involved are large, so the trials also have a better chance of showing up any unexpected adverse side effects. Right. So we have such a wide range of people, such a wide range of variables, so many different kinds of people, so many different kinds of variables. So side effects are almost guaranteed to be spotted, and if there are no side effects, then we truly know that this drug is effective, it is safe, no side effects will happen, no unexpected, no unacceptable side effects. But of course, all people are different. So no matter how many people you try, there is always going to be an outlier. There's always going to be an anomalous result. That is just how it works. Phase three trials are used to confirm that, to a certain degree, of course. Um, so the point of the phase two trials, the point of the larger sample size, is to have a better chance of showing up any unexpected adverse side effects. That is the point of these trials. Patients are randomly allocated to receive the new medicine or the control slash placebo. Uh, Data on effectiveness, side effects, or and other information are collected and assessed to see if there's any system. Okay, I need to pay attention for this. So we were talking about phase three and what happens during the phase three trials, right? So, um, first of all, they're randomly allocated to make sure that there's no placebo effect. Done. Randomly allocated and put into different groups, where one group is going to have one group is going to have. I need to make sure this is sometime because I was feeling that it isn't. Okay, I'm still using my data. Okay. Okay. So, why is there sunlight here, man? Hmm, so damn bright. Okay. Oh wow, this is much colder. Okay, okay. So what happens during the phase three trial? Now they're actually telling us. So, patients are randomly allocated. Placebo is removed to the highest degree that they can, and data on effectiveness. So what's collected, and what are the results of these trials? You have data. You have data on effectiveness. You have data on side effects, aka safety. Other information are collected, aka let's say how easy is it to ingest or to excrete these medicines, right? Other information could be specific side effects that might happen in older people that just got discovered. We didn't use a sample size this old, so now we're truly seeing what's different when we use a sample size this old. It's like people who are very fit don't seem to be that effective. Like the medicine doesn't seem to be that effective on that people. These are some data on effectiveness, right? Some data on side effects, and some data on other information. um, what's the other thing that could be looked into? Stable. Yeah. So like different people, different environments that the medicine is in, right? So that's another thing. So all of these, it is truly such a vast amount of information and such an interesting amount of information that they just put into one sentence here. It's collected and assessed, and it's assessed. So we study so much about evaluation. Why? So that we can look into this information and our brains can connect the dots and assess. Can we see any statistically significant differences between the new medicine and the placebo or the currently available drug? These are the three things that we're testing truly. So there's the new medicine, there's the placebo, or is the important part. I'm sorry, or is the important part, the currently available drug. So this falls into one group. Okay, so this is exactly what we've been studying evaluation for, assessing for. That's why there's so many 9 markers, 6 markers on these kinds of questions. Because truly, when we become biologists, when we become doctors, the hardest part is, or can we find statistically, looking at graphs, looking at tables of data, statistically find significant differences between new drug and placebo slash currently available drug, to spot that is it truly worth it to produce hundreds of millions of thousands of billions of dollars worth of medicines? Is this worth it? Okay, that is why this is truly. I mean, that tells us more about us than the medicine drug testing itself. So continuing on. Uh, that was a tangent. It is difficult to achieve a complete set of results in clinical trials because so many patients stop taking the medicine for various reasons. Some do not take it regularly. That makes sense. I don't take my medicines regularly. That's for sure. um, God damn it. So that's why it's complete. That's why it's difficult to achieve it because people don't take medicines regularly. That's the truth. It's human nature to not do things consistently, and this is one of those things. They don't have the medicines regularly, so you can't get a complete set of results. AKA, that's why the results aren't fully. You can't use them fully because they're not the complete. That's not the complete course. If we're doing an antibiotic, we need to complete the course. If people don't complete the course, then I can't give you the full result. It's not the full result. I can't make the best, table of data. I can't make the best comparisons. I can't make the best side effect awareness. All these awarenesses all these datas and all these awarenesses that I'm telling you, I'm trying to find with the results of the phase three cannot be made if the phase three is not done properly by the patients themselves, right? So there is also quite a bit excuse me, quite a bit of reliance and dependence on the patients themselves. If you can hear the children, I would be really happy because it's so adorable watching this. Okay, anyways, in some trials the new drug or drug combination is so successful that the trial is halted early. It is so successful. The results are so good. There are no side effects which are bad. There are no unexpected things. It is just so well made, especially if the size of the sample is so high. It is there seems to be nothing going wrong. So we don't waste time. We halt the trial early. Perhaps we halt the let's say this is the phase two trial. We halt the phase two trial early. Bam, hit it to phase three. Get it into the best kind of testing. Get it made as soon as possible. So, if the evidence shows that a new treatment is so particularly effective, it becomes unethical to deny the new treatment to the patients for receiving the old treatment on the placebo. That is insane. That is. I wonder. I'm gonna ask Ms. Durga, is there any examples of this, of a drug or of a medicine being so successful that it is actually unethical to give the people who are given the earlier trial or earlier or like the currently best medicine, or placebo? It becomes unethical to give them that. This is the best case scenario, right? Okay, if a new drug is found to be safe and effective in the phase three trials, this is the normal case scenario, and it's found to be safe, so it succeeds. It succeeds the phase three trial. This is the last phase three trial. We were talking about phase three trial over here. If it succeeds that phase three trial, then what happens? The pharmaceutical company will try and get approval for the medicine to be sold. Now we have gone into finally the economical side of this. The pharmaceutical company comes in, tries to get the medicine sold. Sorry, rather, they will try and get approval from who? From people to get the medicine sold. Okay, so now we have received the second pair of the second group or third party to help us out. First, it was the regulation authorities. Now it's going to be the pharmaceutical company. All the data submitted and all the data is submitted. All the data submitted are assessed to evaluate the beneficial effects against possible harmful effects of the medicine before a decision is made on whether or not to grant a license for the product, aka even more evaluation from the pharmaceutical company. What are we evaluating? If this medicine should be given a license, if this medicine should be on the market, if this medicine should truly be a product that people are using. Why are we evaluating? Well, if we are given data, and even after all these trials are successful, there is still perhaps, like, let's say there is a pro and a con to this medicine. The pro is great, but the con exists. At this point, the pharmaceutical company still needs to assess the beneficial effects and the harmful effects of the medicine before it's again given onto the market. Such a license that is needed before a new drug can be put on the market, or an existing drug can be used to treat a different disease. AKA, what is this license that we're talking about? Well, we were right in the first part that the license allows a new drug to be put on the market. But a license also has another use of actually getting that drug to, if we refine the drug to make it better suited for another disease, then after the phase one, phase two, phase three trials, let's say that drug, that chemical, can bind to different proteins. It can bind to different enzymes. We spot that they have the structure to do that. So that means that if it has the structure to do that, then we can again do the phase one, phase two, phase three trials, get the license for it to actually switch its function to a different disease, like make that known to the products, change the product name into a different thing. I have to go 10:30. No, I have to go 10 o'clock. I have 10 minutes. Lesson one, lesson two, lesson three gone. Lesson four. Okay. Fire. Okay, let's get this. Every once a new medicine is being used to treat patients, trials continue. Even after the new medicine, even once a new medicine is being used to treat patients, trials continue. So, let's say medicine has gone through the last trial. Oh my God, they got the license. They are on the market. They are considered a medicine now. They're not longer a compound. That is, what did they use? What term did they use? Yeah, it's no longer considered a new compound. It's now considered a useful medicine, and it's being used to treat people now. Okay, but however, trials still continue. Medicine will be monitored for safety and effectiveness for as long as it's going to be used, even tens of years after the medicine has been discovered. People are still getting trials for it, and this sun is still coming on my ass, dude. God damn, relax. Okay, so why are we doing this? Why are we still doing trials after three whole trials are being done? After three whole trials, after so many assessments, after so many evaluations, after so many data is collected, after tens of thousands of people, why are we still testing it as time goes on? Because any adverse reactions suffered by patients are reported. Again, when it is on the market, you will have the greatest sample size. And if you have the greatest sample size, you have the greatest variation in environments. You have the greatest variation in variables, aka environments. So you have the greatest variation in possibly the side effects, the safety, and also the effectiveness, right? So if there's anything that is it's monitored for safety and effectiveness. And if there's any adverse reactions, then it gets reported and recorded to ensure that benefits of the medicine is always greater than the risks. Constant evaluation. I'm constantly seeing, is there any reports coming to me that this is bad, this is killing this person, this is hurting this person, this is helping this person? What is wrong with that person? What is special about that person that it's this instead of all the good things that are happening to the hundreds or the tens of thousands of people that I've tested already? What is special in this person? Is that environment common enough? Now I need to evaluate if that adverse reaction happens in that environment. Is Do I still allow this drug to be on the market? Can I still allow it to be on the market? I need to constantly make evaluations on this. There are so many evaluations involved in testing whether or not a drug should stay on a market or not. Right? There are so many, and so even after the drug is placed on the market, you are going to get reports. You are going to get um, mm, you know you're going to get calls from people about people where the drug is not being useful, and that is and then there are adverse reactions. Perhaps there are adverse reactions in the sense that it's poisonous. It's not just not working, but it's also having a negative effect. This is a pretty alarming thing. So you need to constantly monitor these reports, constantly record these reports to ensure that you can so why are we recording it again? To have a list, to have data, so that you can review it, so that you can evaluate it, so that you can assess it, to see that is the benefits of using this medicine always greater than using the risks? Because the moment you evaluate it and you're like, okay, fine, the risks are greater than the benefits, get it off the market ASAP, right? Here is a diagram summarizing the main stages in standard drug development protocol. We're having early phase research, preclinical testing, clinical trials, regulatory review, manufacturing, and post-market surveillance. So if you remember, the moment it gets its patent, it has 20 years. From what we see here, let's say we take the max here: 6, 7, 7, 14, 14, 16, and continuous onwards. So when you go from early stage testing, you have 5,000 to 10,000 compounds which link onto that specific enzyme. When we go to preclinical testing, we have 250 compounds which seem to be binding to that enzyme, binding to that protein, which is related to that disease process. 250 compounds are doing that. That is a huge difference from 5,000 to 10,000. So 250 are binding and are affecting that specific process. Clinical trials is when now you're testing 5 different chemicals. They seem to be doing the same exact thing on in the environment, in the computer trials, in the simulations that we are putting it in. They seem to be doing the same exact thing. However, of course, there are differences in the variation environments, in the way they are extracted, in the way they are ingested, right? In terms of effectiveness, in terms of how, if I put it for long scale, for long term, is it still acting the same way, right? There are going to be differences between these compounds, and if there isn't, then that's a different case, right? So now, as we're testing it amongst even more and more and more and more and more and more and more people, from phase 1 we're testing it on certain people, phase 2 even more people, phase 3 on even more people, and in phase 2 and phase 3 we're ensuring that we don't even know what compound we're using, because it's we wouldn't want placebo to affect both the doctors nor the patients. We don't want placebo to affect that, because you don't know um, what the mind can affect the body. You don't know if the doctor is when evaluating these results will be biased to one rather than the other, just because he prefers one rather than the other. Even though he says that he is unbiased, there is always a bias, and that is why blind trials. Even if he is the best doctor, there is going to be a bias. Perhaps he doesn't. When he uses the drug, it doesn't work properly. He is going to be biased one way or another, because humans are bad at perceiving how much the mind controls the body. Okay, or rather, controls affects the body. It doesn't control the body, but it affects the body. Okay, and so we used double-blind trials during phase 2 and phase 3 trials, and the number of products that we're using decreases and decreases as the sample size increases. It increases until we reach the regulatory review, where we have gotten the person, the pharmaceutical company, and we are evaluating with them whether or not the pros are better than the cons, whether or not the side effects are too adverse, too bad, to be put on the market, whether or not the variety of environments that the doctors have used in their phase 2s and phase 3s, if the range of variables is good enough to be put them on the market, is it safe enough? We are reviewing that in this regulatory review. We are assessing that. We are evaluating that, and then finally, in when we finally reach marketing, and also regulatory review, the pharmaceutical company could also be assessing: is it cheap enough? Are we making a good enough profit? Right? Let's get a bit political here. You know, perhaps let's get a bit conspiracy theory guy here. Perhaps if a person has found the cure to cancer, that's going to be putting a lot of businesses, or let's say he got found a cure to the common cold. That's going to be putting a lot of businesses, a lot of money out of the pharmaceutical company's hands, right? So perhaps even in the economical um, economical standpoint, right? Perhaps regulatory reviews can deny the deny the compound's right to exist on the market, because it's unethical for the company, or for the pharmaceutical company, to or it's unrational, if we're talking about economical terms, it's unrational for that pharmaceutical company to allow them that drug to be on the market. I'm not supporting them, but this is from an economical point of view. This is going to be who makes the most money this is a whole different thing. When we're talking about people who are developing drugs, these are people who are testing if these drugs can work the best, if these drugs can help the most people with the least amount of adverse side effects. But when we look at when the pharmaceutical company gets these drugs on their hands, then this is about whether or not this can make us the most money. They don't really care about how good the drug is, how effective or how safe, or how easily the drug is used. They don't care about that. Of course they would, because those things will definitely affect the sales of the product. But of course they also care about the economical side. How cheap is it, right? If it's too effective, then that might be putting the pharmaceutical company out of business. So they won't put it on the market. They won't give it the license. They won't let it they won't let it start being marketed. They won't let it start being manufactured because it's too expensive, or maybe it's putting too much money out of the company's hands, although it's helping the company. So that's going to be scaled up, and of course this is going to be done in years. So of course the most time is going to be put into this. The second most time is going to be put into phases, but each phase is going to be much less. 1 year is going to be put into preclinical testing, aka the animals, aka the plants, the cells, the tissues, all that stuff. Um, no, I don't know why I said plants, not plants. And then the manufacturing is 0.5 to 2 years, and the continuous is of course after you have the drug put on the market, after you get the crazy, 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 crazy sample size, you're gonna start getting different kinds of results, different kinds of adverse reactions, different kinds of adverse effectivenesses, and as a result, people are going to stop using it, or maybe there's going to be decisions that or evaluations that have to be made. So that is going to be the post market surveillance, um, allowing this drug to either stay on the market or be refined even further to be used as a better drug. XYZ, it's continuous, it's going to happen over a long period of time. That's going to be it. That is going to be it for this video, for this chapter, for my life. Wait, hold on. Let's not do that. No? Okay. Uh-huh. I'll see you. Have a nice day.